Dear students, welcome to Chapter 6, Part 1. This section of the book deals with DNA replication. This may seem a very simple and straightforward process to understand, but it does cause a lot of complications for students because of the detail involved. To do well on this material, students have to remember some basic rules about DNA replication. These are explained in Module 1, which follows next. The first learning outcome is therefore to understand the rules by which the enzymes which copy DNA abide by, and also to understand the process of proofreading as it pertains to DNA replication. Thus, we shall be addressing these two important concepts from the Chapter 6 word bank. Inside living systems, new DNA is made from old DNA. So this requires the assistance of enzymes, as we've learned in previous chapters. The most important enzymes are named after the process that they perform. So all enzymes in all living things which copy DNA from existing DNA are called DNA polymerases. So the word polymerase means to polymerize something. And in this case, those are the nucleotides used for making fresh DNA. There are some minor and major differences in the various enzymes which we have recovered and classified from prokaryotic organisms versus eukaryotic organisms. In general, some of these characteristic differences are in the ways that they copy the DNA and how they repair DNA, as well as some very specific functions which we'll address much later in the chapter. The number of such DNA copying enzymes discovered so far has increased over the last decade or so. In prokaryotes, we have identified 5 separate classes of DNA polymerases. They differ in their speeds and their ability to proofread the DNA, as well as other properties which are not important to us in this chapter. Likewise, in eukaryotes, we have identified 16 different DNA polymerase, and these are also classified according to various properties, as well as the locations within the cell at which they operate. Because there's so many varied DNA polymerases, we will focus our attention first on several important properties of the most common DNA copying enzymes found when chromosomes are replicated. The actions of the main DNA polymerase enzymes during DNA replication cannot begin unless these 3 criteria are first satisfied. The first of which is the need to have a template strand available from which to read the information needed to make the new DNA strand. The second requirement is the existence of the raw material, the mononucleotides necessary to compose the new strand. These will be the A's, the G's, the C's, and the T's, and these are normally available in the environment immediately adjacent to the enzyme. And the final requirement is the existence of a 3' free end to which the new incoming nucleotides would be covalently bonded using phosphodiester bonds. This is by far the most confusing component of these requirements, and we shall explain that shortly. One further outcome of the action of DNA polymerase is indicated here. Students are familiar with this. Whenever a T base is encountered on the template strand, the enzyme adds an A opposite the T on the new strand. Whenever a is encountered on the template strand, it adds a T opposite. Likewise with G and C. This slide summarizes all the rules in one place. So rule number 1 is the requirement for the existence of a template, which will then be copied. The second rule is the existence of free nucleotides, the raw material for making fresh DNA. And finally, the requirement for an existing 3' end on a strand of DNA that will be then copied further by these enzymes. And finally, the incoming nucleotide is selected by the enzyme based on the existing nucleotide on the template strand. If this was a T, an A would be added here. The polymerization process would continue as long as there was a template available for the enzyme to read. A further critical emphasis must be made here for the students to adhere to. As you can see, the 2 strands of DNA, the bottom strand, the template strand, and the newly made strand on top, both have an orientation. The bottom strand is read in the forward direction. That's 3, 4, 5. All DNA is read in the forward direction by DNA polymerases. In the case of the newly made strand, that is made backwards. So 5, 4, 3, and it will continue to be made backwards as long as there's template available below to copy. Please do not forget this fact and try to remember this in future diagrams as we progress through this chapter. If we delve a little deeper into the information, we will see some important concepts reappearing in this chapter, which we learnt in chapter 5 and then also in chapter 2. In this particular diagram, the template strand is on the right-hand side of the page, and the newly made strand is being formed on the left-hand side. Regardless, all the rules discussed previously are being adhered to by the DNA polymerase. As you'll discover later, DNA replication is actually performed by a whole host of enzymes working together in a very coordinated fashion. And as we learnt in the previous chapter, whenever proteins work together in a coordinated fashion, we can refer to that as a machine. So in this case, DNA replication is not performed by a solitary enzyme, DNA polymerase, but by a combination of many proteins working in unison, and that is called a replication machine, a DNA replication machine to be more precise. Here one can see a moment in time as an incoming nucleotide is being polymerized to an existing 3' end on the newly made strand. So the requirements are that we must have a free 3' end on the existing nucleotide that is part of the growing strand. And that is satisfied here because as we bring in the new nucleotide, in this case a C nucleotide, we find that it has the necessary orientation to be polymerized to the existing new strand. The reason why we're adding a C is because that is the only nucleotide which would fit stereochemically across from a G. One further observation is that we also satisfy the backward direction in which DNA is made. Here we have the 5' end of the molecule, and here we have the 3' end of the molecule. And as soon as this nucleotide goes in, we'll have a new 3' end. So the new strand is being made backwards, 5, 4, 3. And as it says at the bottom of this slide, errors in incorporation are pretty rare, so DNA polymerase is a pretty reliable enzyme. It is said to have high fidelity. Even though the enzyme makes few mistakes, it does make mistakes on occasion. It puts in the wrong base opposite a particular base in the template. In this case, it doesn't leave the wrong base in position. It has the ability to recognize that something is wrong in the chemistry. It backs up and removes bases from the DNA and starts again. This ability to stall, move backwards, and reproduce is called proofreading. And this proofreading ability reduces the error rate to an acceptable level when copying DNA. It's important again to reiterate the fact that when the enzyme is making DNA, it does so backwards. Thus, it is reading the template strand forwards, but making the new strand backwards. However, during proofreading, it reverses direction. It stops and moves backwards. It doesn't polymerize any new DNA nucleotides. In fact, it removes old nucleotides from the DNA new strand, therefore able to correct its mistakes. And that is further shown in this slide, where the green circle represents the DNA polymerase, and you can see that it's incorporated the wrong base in this case, and it backs up to remove the wrong base and then inserts the correct base. With respect to the functionality of the enzyme itself, we can see that it has a couple of active sites, each responsible for a different function. The site here, labeled P, is responsible for making new DNA, the polymerizing function. However, there is a second active site on the enzyme called the proofreading site, labeled E. And should that site be necessary, it then has the ability to remove nucleotides, which it added using the P site. The inclusion of this slide is for education purposes only. Here we have a hypothetical situation which does not occur in real systems on the left-hand side, but we have the actual situation which does occur in living systems on the right-hand side. What this slide is trying to convey is that the orientation of the DNA is very important when it comes to which side of the incoming nucleotide carries energy. As we've learnt in this class, all molecules have a shape, and that shape is important when they interact with other chemistry within the cell. Here, the situation is no different. If we examine the hypothetical situation, the incoming nucleotide brings its energy on the left-hand side. So the pyrophosphates, the two pyrophosphates here, and the first phosphate group are on the left-hand side of the incoming nucleotide. If that nucleotide was added to the growing DNA strand on its left, the energy is supplied by the preceding nucleotide, indicated here. So the green base nucleotide has 3 phosphates. When the red nucleotide enters and is polymerized to the preceding green nucleotide, the energy is obtained from the breakage of this bond here. Now everything is fine as long as the chain continues to grow without any mistakes. However, if there is a mistake and the enzyme has to remove the red nucleotide, then there's a problem. Removal of the red nucleotide leaves us with the preceding green nucleotide now missing its pyrophosphate. The energy has all been used in the previous step. The incoming nucleotide, the correct nucleotide, in this case the blue one, cannot be bonded because the preceding nucleotide does not have the necessary energy in the right place for the formation of that phosphodiester bond. Therefore, proofreading would not be energetically viable had the nucleotide been added to the 5' end of the growing DNA. Let's now concentrate on the right-hand side of this slide. This is the solution that nature has evolved. Here, the growing DNA strand is only added to at the 3' end. Here, the incoming red nucleotide is added to the strand at the 3' end, and that energy is supplied by the pyrophosphate of the incoming nucleotide. So energy is brought to the bond by the incoming nucleotide. The chain will continue to grow as before unless a mistake is made. If the polymerase makes a mistake, then proofreading activity is activated. Here the wrong nucleotide is excised, and now we have to bring in the correct nucleotide. Once again, the blue nucleotide is the correct nucleotide, and that has to be bonded to the preceding blue nucleotide. Here the energy is brought in by the incoming nucleotide. So each incoming nucleotide brings its own packet of energy. And that energy is used to now bond correctly the blue nucleotide to the preceding blue nucleotide. And this can be repeated indefinitely as long as the incoming nucleotide brings its own packet of energy. So the main difference is, where is the energy located? On which side of the incoming nucleotide? Here the energy is too far away from the bond. Here the energy is close to the bond and can be utilized. Students should understand this before proceeding. This concludes the first module. You should be in a position to now describe the rules by which DNA polymerases replicate DNA, and also have a good clue as to the proofreading process. In Module 2, we turn our attention to the actual replication of DNA as it occurs inside a living cell. The module learning outcomes are described here. At the conclusion of this module, the structure of a replication bubble needs to be understood, and the component parts that make up the replication bubble need to be defined. Also, the direction in which the replication forks travel along the DNA has to be understood and relayed. And finally, using illustrations, the orientation of new fragments of DNA added at the replication fork have to be correctly deciphered. Examination of the Chapter 6 word bank shows us that most of the words apply to this module. Historically, for a period of time, there were a number of competing models trying to decipher how DNA was replicated. Since then, our understanding has focused on the main type of DNA replication that appears to be taking place inside living things, and that is called the semi-conservative model of DNA replication. As illustrated here, replication results in the separation of the 2 strands of DNA in a DNA double helix and the generation of a fresh new strand opposite each of those original DNA templates. This process continues from generation to generation, resulting in the preservation of the original parental strands and the formation of fresh new strands. In all living organisms, the replication of DNA begins with the generation of a replication bubble at a special location predetermined on the DNA where replication begins. And these predetermined locations are known as origins of replication. So here we have a chromosome with 3 origins of replication. At the appropriate time, when replication is initiated, bubbles form at these locations on the DNA helix. And it is within these bubbles that DNA synthesis takes place using the semi-conservative mode of DNA replication. The bubbles then increase in size, growing towards each other until they merge, and the result is 2 identical chromosomes, each with 1 original strand of DNA in brown, and each with 1 new strand of DNA in black. If one looks closely, each bubble has a left side and a right side. These left sides and right sides of every bubble are referred to as replication forks. So this bubble in the center has a left replication fork, which is traveling to the left, and a right replication fork, which is traveling to the right. The situation is complicated by one of our rules that we discussed earlier, that DNA can only be synthesized in one direction. So here we have just the right side of one replication bubble, and as the bubble is growing towards the right, we have the generation of new DNA indicated in red on both the upper parental strand and the lower parental strand in orange. According to the rules, the top strand has a 5' end on this side, and the bottom strand has a 3' end on this side. According to the rules that we discussed earlier, DNA can be grown backwards. So 5, 4, 3, and new nucleotides can be added here as the 2 parental strands are unwound, and the red strand will continue to grow into that space. However, the top strand is going to be made forwards if we continue with this hypothesis. So 3, 4, 5. Now DNA cannot be added to the 5' end for reasons we discussed earlier. Therefore, the 5' end of the top strand cannot be grown in this direction. It can only be grown in the reverse direction. This presents living cells with a conundrum. The solution to this conundrum is quite elegant, as we'll now see. The solution is to continue to make the bottom strand in the forward direction as that bubble continues to grow to the right, and to make the top strand backwards. As the bubble opens, new primers are laid down, and it is then grown in the reverse direction until it joins a pre-existing strand of DNA. So these fragments that are made discontinuously as the bubble opens up are known as Okazaki fragments, after the Japanese scientist first discovered these. Conversely, if you look at the left side of the bubble, at the left replication fork, the situation is flipped upside down. The top strand is made in a continuous direction, and the bottom strand is made in a discontinuous reverse direction compared to the right-hand side. So as the bubble grows over time, new DNA is added as indicated in red on the bottom half of this figure. The bottom strand on the right fork continues to just extend into the unwinding DNA, as does the top strand in the left fork. The situation is more complex with regard to the newly synthesized fragments in the top half of the right fork and the bottom half of the left fork. It is only here that Okazaki fragments are found temporarily until these pieces of DNA are joined together by special enzymes. If we zoom in onto the right fork and the top strand, we see that the situation presents itself with a biochemical challenge. As we were alerted to by the rules in Module 1, for DNA synthesis to take place, the DNA polymerase enzyme, it needs a template and a 3' end. However, as the fork opens up, there is no 3' end within that fork to which the DNA polymerase can add new nucleotides. So essentially, DNA replication cannot continue within a replication bubble. This is remedied by the existence of another enzyme called RNA primase, which adds a RNA primer, indicated in blue, to DNA at the replication forks. This very short piece of RNA then provides the DNA polymerase with a 3' end to which it can extend. The DNA polymerase continues to extend the DNA until it encounters another primer from a preceding fragment of DNA. The particular form of DNA polymerase here can no longer continue its job. It falls off the DNA, and another enzyme, another form of DNA polymerase, settles on the fragment at the gap and eats away the RNA primer until it's removed. Yet another enzyme comes in to close the nick, forming a continuous fragment of DNA. The same thing would happen here as another piece of DNA is grown into this region from the right-hand side. So this solution, involving the switching and swapping of multiple enzymes, is nature's way of overcoming the requirements that DNA can only be extended in the backward direction. So in reality, in natural systems, DNA replication cannot begin unless RNA is first deposited on the DNA template. This leads to the conclusion that within a cell, RNA is the master and DNA is the slave. As mentioned earlier, DNA replication involves a machine, and here we are looking at the components of the machine in a two-dimensional view. The top strand, the one that's synthesized in the direction of the opening replication fork, is known as the leading strand. So that's called leading strand synthesis. The bottom strand, which presents us with a problem, is made in the reverse direction, and that is known as the lagging strand. It's on the lagging strand that we find Okazaki fragments and RNA primer dispersed in regular repeating patterns. There are many proteins involved in a replication machine, but the main ones are listed here as a list of 7. So DNA polymerase 1, DNA polymerase 3, DNA ligase, DNA helicase, single-strand binding protein, DNA topoisomerase, and RNA primase. Students need to learn this list and the function of each one of these. When the same replication fork is viewed in 3 dimensions, the machine takes on this very compact shape. The 2 DNA polymerases are actually held together by a complex series of proteins, all forming a giant machine. The best way to learn this is through animations. The book does supply a number of animations on DNA replication, and there are nice ones on the internet that the students should view. Once the student has done that, then these learning module outcomes can be then tackled. The final module of this video looks at a type of DNA polymerase known as telomerase. This enzyme is only relevant to linear chromosomes, such as eukaryotic chromosomes. Once this module is concluded, students should be able to describe how this enzyme, telomerase, helps some cells with linear chromosomes replicate their chromosome ends. The evolution of telomerase in the first place has to do with a shortfall in the regular DNA synthesis enzymes. In eukaryotic cells, these enzymes are unable to reach the extreme ends of linear chromosomes. This results in the systematic shortening of the ends of every chromosome following a round of DNA replication. For many cells in the body, most somatic cells, this enzyme is turned off early on in the life of that cell. So over a period of time, many tissues in our body eventually result in their chromosomes being shortened until a critical location is reached and the cell line then dies. This process therefore places a limit on the longevity of human lifespans. On the contrary, germ cells, which have to be passed on to future generations, cannot afford to have their chromosomes shortened every generation. So it is inside these cells that telomerase has its function. It is designed to extend the length of these chromosomes to keep their length stable and prevent shortening. Incidentally, the reason that cancer cells can grow indefinitely is the result of them activating the telomerase gene that they possess. This makes cancer cells immortal. How does the enzyme telomerase accomplish its task? Well, the answer lies in the fact that the enzyme works with RNA. There's a piece of RNA that is embedded inside the enzyme, and the two components, the protein and the enzyme, work in harmony by attaching to the extreme ends of repetitive telomere sequences. So in this figure here, the brown areas represent the extreme ends of our chromosomes. These are the ends which have to be extended in order to stop the chromosome from shortening. If the shortening continues into the gray areas, then that cell becomes unviable and dies. So what the telomerase does is it uses the RNA component to bind to complementary DNA sequences and form a hybrid molecule. The activity of the telomerase then extends the 3' end of the DNA in the direction in which nature intended. The enzyme then hops along multiple times, up to 200 or 300 times, extending the single strand of DNA at its 3' end. This permits the normal DNA replication mechanism in identifying and laying down primers against the extended repeats. So DNA polymerase will now use RNA primase to lay down a primer and then extend itself back to form a double-stranded region. So the double-stranded region prevents the shortening of the chromosome in this very complicated hopscotch manner. Students are advised to view the accompanying video for this chapter. Once the video has been digested, then students should return to be able to describe how telomerase enzyme activity helps replicate their chromosome ends. This concludes this video on DNA replication. Thank you.